Welcome to part 1 of our 11-part series on intracellular structures, developed specifically for the European Society of Human Reproduction and Embryology Clinical Embryologist certification. This first session lays the essential groundwork, the cellular foundation upon which every concept in reproductive medicine is built. Before we can begin to understand oocyte maturation, spindle dynamics, fertilization, or embryo development, we need to be fluent in the language of the cell itself. Think of this session as your orientation into the embryologist's true workplace. Every observation you make at the microscope, every clinical decision you take in the laboratory, ultimately traces back to what is happening inside cells at the subcellular and molecular level. The European Society of Human Reproduction and Embryology Competency Framework is explicit about this. Proficiency in cell biology is not optional background knowledge. It is the foundation of clinical reasoning in assisted reproductive technology. Over the 20 slides that follow, we will move systematically from the broadest principles of eukaryotic cell organization down to the specific intracellular architecture of oocytes, sperm, and early embryos. By the end of this session, you will have a clear, integrated map of how cells are organized and, crucially, why that organization matters so profoundly for the outcomes we care about most. Let's begin by understanding exactly why cell biology sits at the very heart of everything we do in the in vitro fertilization laboratory. Every in vitro fertilization outcome, every pregnancy, every live birth, every disappointment, begins and ends at the level of the cell. This is not hyperbole. It is the central reality of clinical embryology, and it deserves a moment of genuine reflection. Consider what we actually do in the laboratory. We retrieve oocytes, single cells approximately 120 micrometers in diameter. We inseminate them, or we inject a single sperm using intracytoplasmic sperm injection. We watch for fertilization, a process defined by the appearance of two pronuclei, each representing the successful completion of intracellular events that take place within hours. We culture embryos for 5 to 6 days, observing their cleavage, compaction, and blastulation, all of which are expressions of intracellular machinery either performing precisely as it should or failing to do so. What you observe under the microscope, the size and symmetry of blastomeres, the presence of fragmentation, the quality of the inner cell mass, the granularity or clarity of the cytoplasm, is a morphological window into what is happening inside those cells. Understanding cell organization gives you the interpretive framework to look at these morphological features not as arbitrary aesthetic criteria, but as biologically meaningful signals about intracellular function and developmental competence. The European Society of Human Reproduction and Embryology Competency Framework reflects this directly. Clinical decision-making in embryology is grounded in cell biology. Understanding the cell is not an academic luxury. It is a professional necessity. With that motivation firmly established, let's map out exactly what we'll be covering across this session. Before diving into the content, let's take a moment to orient ourselves. A clear map of the session will help you organize new information as we build it layer by layer. And in a topic this rich with interconnected concepts, that orientation pays dividends. We will open by defining what makes eukaryotic cells fundamentally different from prokaryotic ones. The answer comes down to a single organizing principle: compartmentalization. From there, we will carefully distinguish 3 terms that students frequently conflate: cytoplasm, cytosol, and organelles. These distinctions are not semantic pedantry. They reflect real differences in biochemical function and clinical significance. We will then examine 2 categories of organelles: membrane-bound structures, which maintain their own internal microenvironment, and non-membrane-bound structures, which are equally essential but organized differently. This will lead us naturally into the cytoskeleton, the architectural scaffolding that links all of these components, positions them within the cell, and enables the physical movements required for cell division. In the second half of the session, we apply all of this to the specific cellular contexts that define our clinical work: the oocyte, the sperm, and the early embryo. We will see how intracellular organization directly predicts developmental competence, and how assisted reproductive technology procedures at every step impose measurable stress on these architectures. Think of the entire session as building a biological narrative, from first principles through to clinical application. Now let's set our specific learning targets so you know exactly what you should be able to do by the time we finish. 5 learning objectives define the scope of this session, and they are deliberately sequenced from conceptual understanding through to clinical application. Walking through them explicitly is worthwhile, because understanding what you're expected to achieve shapes how you engage with the material. The first objective asks you to define and distinguish cytoplasm, cytosol, and organelles. This sounds straightforward, but these terms are used imprecisely in clinical literature all the time, and that imprecision creates real conceptual confusion. We will be rigorous about these definitions throughout. The second objective introduces compartmentalization as the organizing principle of eukaryotic life. You should be able to explain not just what compartmentalization is, but why it matters, what advantages it confers, and what happens when it breaks down. The third objective requires you to identify the major membrane-bound and non-membrane-bound organelles and articulate their roles. Think of this as building your intracellular vocabulary. The organelles are the characters in every cell biology story we tell. The fourth objective bridges cell biology and reproductive medicine directly. You will describe how intracellular architecture supports the specific processes of oocyte maturation, fertilization, and early embryo development. This is where basic science becomes clinical science. And the fifth objective asks you to relate disruptions in cell organization to impaired developmental competence and assisted reproductive technology outcomes. This is the integrative, analytical level, the level at which European Society of Human Reproduction and Embryology certification questions are pitched. Can you observe an embryological finding and reason back to its likely intracellular cause? By the end of this session, you should be able to do exactly that. With our objectives in place, let's begin building the foundation by examining the eukaryotic cell as a system. Eukaryotic cells are defined by one overriding architectural feature. They are membrane-enclosed compartments within a larger membrane-enclosed compartment. That layered organization, membranes within membranes, spaces within spaces, is what distinguishes them from prokaryotes and what makes the extraordinary biochemical complexity of human reproduction possible. Let's think about what this actually means in practice. Each compartment, the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and endosomes, maintains its own distinct internal microenvironment. Different hydrogen ion concentrations expressed as potential of hydrogen. Different ion concentrations. Different redox states. Different enzymatic repertoires. This is not incidental. It is functionally essential. It allows incompatible biochemical reactions to occur simultaneously in the same cell without interfering with each other. Anabolism proceeds in one location while catabolism proceeds in another. Oxidative reactions are physically separated from sensitive deoxyribonucleic acid. Hydrolytic enzymes that would destroy cellular proteins are safely sequestered in lysosomes. Here is a question worth sitting with for a moment. Why does this matter so specifically for the oocyte? Because the oocyte is not just any eukaryotic cell. It is the largest human cell, approximately 120 micrometers in diameter, roughly 10 times larger than a typical somatic cell. It is also one of the most spatially complex, with distinct cortical and interior domains that must be maintained in precise relationship to each other throughout maturation and fertilization. The cytoskeleton, the structural framework connecting all compartments, is not merely supportive in the oocyte. It is a primary organizer of developmental polarity. Spatial and temporal coordination of all these compartments is what makes cells work, and in oocytes, it is what makes reproduction possible. Why? Compartmentalization is the most important architectural innovation in the history of cellular life, and understanding why it exists will help you understand virtually every aspect of cell biology we cover in this series. Let's work through the logic carefully. The first and most fundamental reason for compartmentalization is the need to run incompatible biochemical reactions simultaneously. Consider anabolism and catabolism, building molecules up versus breaking them down. If these processes occurred in the same unpartitioned space, they would be in constant competition, each reaction product immediately becoming a substrate for the opposing pathway. By segregating them, anabolism largely in the cytosol, catabolism in lysosomes. The cell achieves an efficiency that would otherwise be impossible. It is rather like having separate assembly lines running in parallel in a factory, rather than trying to assemble and disassemble the same product at the same workbench. The second reason is concentration optimization. Each compartment can maintain the precise local concentrations of enzymes, substrates, ions, and cofactors required for its specific reactions. Mitochondria, for example, maintain a steep proton gradient across their inner membrane. Without that confined space, the gradient simply could not be sustained and adenosine triphosphate production would fail. Third, compartmentalization enables precise spatial and temporal control of signaling. When a calcium wave propagates across an oocyte at fertilization, it is not random diffusion. It is the controlled release of calcium from a specific subcellular compartment, the endoplasmic reticulum, in response to a precise trigger. The architecture enables the signal itself. Fourth, and critically for gametogenesis, compartmentalization protects deoxyribonucleic acid from cytoplasmic reactive oxygen species. The nuclear envelope is not merely a structural feature. It is a protective barrier separating the genome from the oxidative environment of the cytoplasm, where reactive oxygen species are generated as metabolic byproducts. In oocytes specifically, compartmentalization is essential for maintaining meiotic arrest over years or decades, for organizing cortical granules in their precise subplasmolemal position, and for orchestrating the calcium signaling cascade at fertilization. Disrupt the compartments, and you disrupt the process. Now let's get precise about the terminology we use to describe these intracellular spaces, starting with three terms that are all too often confused. The confusion between cytoplasm, cytosol, and organelles is one of the most common and consequential conceptual errors in cell biology education. These are not interchangeable terms, and using them interchangeably produces genuine misunderstanding about where cellular processes occur and why. Let's build clear definitions. Cytoplasm is the broadest term. It refers to the entire region between the plasma membrane and the nuclear envelope. Everything inside the cell that is not inside the nucleus is cytoplasm. That means cytoplasm includes organelles, the cytoskeletal network, dissolved molecules, and the fluid phase in which all of these are suspended. When you describe a vacuole in an oocyte as a cytoplasmic inclusion, you are using the term correctly. You mean it is located within the interior of the cell. Cytosol is the specific aqueous, gel-like fluid phase of the cytoplasm. It is what remains if you were to remove all of the organelles, cytoskeletal filaments, and membranes from the cytoplasm. The cytosol is the medium in which organelles are suspended, and it is the site of glycolysis, amino acid synthesis and metabolism, and free ribosome-based protein synthesis. When we talk about cytosolic calcium concentration or cytosolic potential of hydrogen, we are referring specifically to conditions in this fluid phase, not in organelles. Organelles are the specialized subcellular structures with defined functions. Some are membrane-bound, the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and endosomes, and maintain their own internal environments. Others are non-membrane-bound structures like ribosomes, centrosomes, and the nucleolus, which are equally essential but organized differently. Why does this matter clinically? Because when we describe a granular cytoplasm in an oocyte or note cytoplasmic vacuoles, we are making observations about specific intracellular spaces. Understanding whether a pathological feature is in the cytosol, within an organelle, or at the cytosol to organelle interface determines how we interpret its significance and what mechanism might underlie it. With our terminology firmly established, let's now take a structured tour of the membrane-bound organelles. Here we have the complete roster of the major membrane-bound organelles, each one a highly specialized compartment with a precisely defined role. The table on this slide gives you a concise overview, and we'll work through the entries systematically because each will reappear throughout this series in the context of gamete and embryo biology. The nucleus is the command center, the repository of genetic information, the site of transcription and ribonucleic acid processing. In the oocyte, the nucleus undergoes a dramatic transition during maturation. The germinal vesicle, the large, visible nucleus of the immature oocyte, breaks down as meiosis resumes, leading to chromosome condensation and spindle formation. The nucleolus within the nucleus is the site of ribosomal ribonucleic acid synthesis. Interestingly, the nucleolar precursor bodies visible in zygotes after fertilization are non-functional initially, reflecting the transcriptional silence of the very early embryo. Mitochondria deserve special attention in the reproductive context. These organelles perform three critical functions: adenosine triphosphate production via oxidative phosphorylation, calcium buffering, and the regulation of apoptosis. In oocytes, mitochondria are present in the hundreds of thousands, the highest mitochondrial content of any human cell type, and they are maternally inherited. The mitochondrial population is not renewed between fertilization and the early cleavage stages, meaning that the quality of the oocyte's mitochondrial pool at retrieval is a key determinant of embryo energy metabolism throughout pre-implantation development. The endoplasmic reticulum performs two jobs simultaneously. It is the site of synthesis and processing for secretory and membrane proteins, and it is the primary intracellular calcium store. In oocytes, the cortical endoplasmic reticulum network is the source of the calcium oscillations triggered by sperm-derived factor at fertilization. You cannot understand egg activation without understanding the endoplasmic reticulum. The Golgi apparatus processes and sorts proteins, directing them to specific destinations including lysosomes, which carry out intracellular digestion and autophagy. Peroxisomes handle fatty acid oxidation and reactive oxygen species detoxification, while endosomes coordinate vesicular trafficking and receptor recycling. Together, these 7 organelle types provide the cellular machinery for energy production, protein biosynthesis, signal transduction, membrane homeostasis, and waste management. Essential as these membrane-bound organelles are, they are only half the story. Let's now turn to the equally critical structures that operate without a lipid boundary. Non-membrane-bound organelles often receive less attention in cell biology curricula than their membrane-enclosed counterparts. But in the context of reproductive medicine, several of them are absolutely central to understanding fertilization, meiosis, and early embryo development. Ribosomes are the molecular machines of protein synthesis, large ribonucleoprotein complexes that translate messenger ribonucleic acid sequences into polypeptide chains. Free ribosomes in the cytosol synthesize proteins destined to remain in the cytoplasm, while endoplasmic reticulum-bound ribosomes produce proteins destined for secretion or membrane insertion. In the oocyte and early embryo, ribosomes are maternally derived, and their activity is directly regulated by the translational control of stored maternal messenger ribonucleic acids, a mechanism we will return to shortly. Centrosomes are perhaps the most clinically relevant non-membrane-bound organelles in reproductive biology. These are the microtubule organizing centers, the structures that nucleate the spindle apparatus during cell division. In oocytes, the meiotic spindle is organized by an acentrosomal mechanism during maturation. However, after fertilization, the sperm-contributed centriole becomes the foundation for the first mitotic spindle in the zygote. This is a remarkable biological asymmetry. The egg provides the cytoplasm and organelle machinery, while the sperm provides the centriole. Sperm with defective centrioles can fertilize oocytes but then fail to organize the first mitotic spindle, a cause of early embryo arrest that is easy to miss without this mechanistic understanding. The nucleolus is a specialized domain within the nucleus responsible for ribosomal ribonucleic acid synthesis and ribosome assembly. Proteasomes are multi-protein complexes responsible for the targeted degradation of ubiquitin-tagged proteins, a critical process for cell cycle regulation, especially at meiotic transitions. Processing bodies and stress granules are cytoplasmic foci involved in messenger ribonucleic acid storage, degradation, and translational control. In oocytes and early embryos, where transcription is largely silent prior to embryonic genome activation, these structures play a key role in regulating which maternal messenger ribonucleic acids are translated when and which are held in reserve. Think of processing bodies as the cell's messenger ribonucleic acid library, holding transcripts until they are needed. Many of these non-membrane-bound structures are maternally inherited and drive all of early development prior to embryonic genome activation, which occurs around day 3 in human embryos. Now let's move to the structural framework that ties all of these organelles together, the cytoskeleton. The cytoskeleton is simultaneously the scaffolding, the highway network, and the contractile machinery of the cell. Without it, organelles would drift randomly, cell division would be impossible, and the cell would lack any capacity for directed movement or organized structure. It is composed of 3 distinct filament systems, each with a unique diameter, composition, and functional specialization. Actin filaments, also called microfilaments, are the thinnest at approximately 7 nanometers in diameter. They are dynamic, rapidly polymerizing and depolymerizing in response to cellular signals, and are concentrated particularly at the cell cortex, just beneath the plasma membrane. Their primary roles include determining cell shape, maintaining cortical tension, driving cytokinesis, and facilitating vesicle movement. In oocytes, actin dynamics in the cortex are essential for polar body extrusion and for the asymmetric positioning of the meiotic spindle near the plasma membrane. Microtubules are the largest filament type at 25 nanometers in diameter, and they are the most structurally diverse in their functions. They form the meiotic and mitotic spindle, the bipolar apparatus that captures chromosomes and segregates them with astonishing precision. They also form cilia and flagella, and they serve as the major intracellular transport tracts along which motor proteins move cargo. The microtubule network in the oocyte is particularly complex, reflecting the enormous range of tasks it must coordinate. Intermediate filaments sit between the two at approximately 10 nanometers, and their primary role is mechanical, providing tensile strength to the nucleus through the nuclear lamins and to the cytoplasm through vimentin and keratins. They are less dynamic than actin or microtubules, but provide the structural resilience that protects the cell from physical stress. A critical and often underappreciated fact: the cytoskeleton is not static. It undergoes rapid, regulated assembly and disassembly in response to cellular signals. What looks like a fixed scaffold is actually a highly dynamic, constantly remodeling structure. This dynamism is what allows the cell to change shape, divide, migrate, and respond to its environment. But it also means the cytoskeleton is vulnerable to perturbations such as temperature change, mechanical stress, and chemical exposure. Notice the 3 filament types illustrated on this slide. Take a moment to appreciate the scale differences between them, as those physical differences reflect their distinct mechanical and functional roles. The cytoskeleton would be a passive scaffold if it were not for the motor proteins that actively move cargo along it. Let's examine these remarkable molecular machines next. Motor proteins are among the most elegant molecular machines in biology. Nanoscale engines that convert chemical energy stored in adenosine triphosphate directly into mechanical movement, walking along cytoskeletal tracks with a directionality and cargo specificity that makes even the most sophisticated logistics system look primitive by comparison. There are 3 major families of motor proteins, each working on a specific cytoskeletal track. Kinesins walk along microtubules, moving toward the plus end, which in most cells points toward the cell periphery. This makes kinesin the outbound carrier in the cellular transport system, moving cargo away from the centrosome toward the plasma membrane. Dynein moves in the opposite direction, toward the minus end of microtubules, which points toward the centrosome at the cell center. Dynein is therefore the return carrier, bringing endocytosed vesicles, signaling molecules, and recycled membrane components back toward the cell center. Dynein also powers ciliary and flagellar beating, a function with direct clinical relevance, since primary ciliary dyskinesia, a condition involving dynein arm defects, can cause male infertility through impaired sperm motility. Myosin works along actin filaments rather than microtubules. Different myosin isoforms contribute to cytokinesis via the contractile ring, maintain cortical tension, and facilitate vesicle trafficking near the cell surface. In the oocyte, myosin-driven actin dynamics are critical for the highly asymmetric cell divisions that produce the first and second polar bodies, divisions that must eject chromosomes without expelling the cytoplasm. Here is a beautiful piece of reproductive biology that elegantly illustrates motor protein function in practice. In the oocyte, motor proteins position the meiotic spindle asymmetrically near the cortex. Think about what this achieves. The meiotic divisions are inherently unequal. The oocyte must expel genetic material in a tiny polar body while retaining the vast majority of its cytoplasmic volume. This asymmetry is not accidental. It is actively engineered by actin-myosin dynamics, pulling the spindle to the cortex. If motor protein function is disrupted by temperature stress, by chemical exposure, the spindle migrates centrally, and polar body extrusion becomes impossible or produces an abnormally sized polar body. In sperm, dynean arms in the axonal microtubule doublets drive the repetitive bending of the flagellum. Absent or defective dynean arms produce immotile or severely dysmotile sperm, a direct cause of infertility that is diagnosable on electron microscopy of the sperm axone. Motor proteins and cytoskeletal dynamics have given us the vocabulary to now examine the extraordinary intracellular architecture of the oocyte itself. The oocyte is, without exaggeration, the most architecturally sophisticated cell in the human body. At approximately 120 micrometers in diameter, large enough to be visible to the naked eye, its intracellular organization has been refined over millions of years of evolution to accomplish one extraordinary task, providing everything necessary to support fertilization and initiate a new organism. Let's map the oocyte's intracellular architecture systematically. Mitochondria in the oocyte are present in numbers that would be astonishing in any other cell context. Hundreds of thousands per cell, the highest mitochondrial content of any human cell type. These mitochondria are entirely maternally derived. The mitochondrial genome passes exclusively through the maternal line. They cluster in characteristic patterns around the nucleus and throughout the cytoplasm, and their distribution during maturation is an active, cytoskeleton-dependent process. Mitochondrial membrane potential, a direct measure of adenosine triphosphate-producing capacity, correlates significantly with fertilization rates, blastocyst formation, and implantation success. When we observe an oocyte with cytoplasmic granularity under the microscope, abnormal mitochondrial clustering is one of the possible contributing mechanisms. The endoplasmic reticulum in the oocyte is not diffusely distributed, but forms a specialized cortical network, a reticular meshwork concentrated in the region just beneath the plasma membrane. This strategic positioning is essential. It places the primary calcium store precisely where it is needed to generate the calcium waves that propagate across the oocyte at fertilization. These calcium oscillations, not a single spike, but a series of repetitive transients over several hours, are required for oocyte activation and the resumption of meiosis II. If the cortical endoplasmic reticulum is disorganized, as seen in some dysmorphic oocytes, calcium signaling may be impaired. Cortical granules are secretory vesicles positioned beneath the plasma membrane in a precisely organized layer. Upon sperm entry, they undergo exocytosis in a calcium-dependent wave, releasing their enzymatic contents into the paravital space. These enzymes modify the zona lucida, the extracellular matrix surrounding the oocyte, cross-linking its proteins to create a hardened, impenetrable barrier to additional sperm. This is the zona reaction, the primary biochemical mechanism of the block to polyspermy. If cortical granules are absent, mislocated, or prematurely released, polyspermy risk increases substantially. The meiotic spindle is positioned asymmetrically near the cortex, enabling the asymmetric cell division that produces the first polar body. And the cytoplasm is loaded with maternal messenger ribonucleic acids and proteins, the biochemical program that drives all of development until the embryonic genome activates around day 3. The oocyte's cytoplasmic quality determines the fidelity of that program. Having examined the oocyte, let's now turn to its cellular partner in fertilization, the sperm, whose architecture could not be more different. If the oocyte is the most complex cell in terms of cytoplasmic organization, the sperm is arguably the most optimized, a cell that has been architecturally stripped down to its essentials with one singular purpose: to deliver the paternal genome to the oocyte as efficiently and reliably as possible. Almost everything about sperm biology represents a trade-off between minimizing mass and maximizing motility and penetration capacity. The head of the sperm contains two essential structures. First, the nucleus, a highly condensed package of deoxyribonucleic acid organized not around histones, as in all other human cells, but around proteames, small, highly basic proteins that package deoxyribonucleic acid at extraordinary density. This proteamine-based compaction achieves a nuclear volume roughly 40 times smaller than a somatic nucleus, reduces metabolic demand, and protects the paternal genome during the journey to fertilization. However, it also means that the sperm nucleus cannot be transcriptionally active, and it must be decondensed. Proteames replaced by oocyte-derived histones after fertilization before the male pronucleus can form. Overlying the anterior portion of the nucleus is the acrosome, a large, Golgi-derived vesicle containing hydrolytic enzymes, including acrosin, hyaluronidase, and various proteases. During the acrosome reaction, triggered by zona pellucida contact, these enzymes are released in a controlled exocytotic event, enabling the sperm to penetrate the zona and reach the plasma membrane of the oocyte. Failure of the acrosome reaction is a significant cause of fertilization failure, and it is not detectable by routine semen analysis. Specialized acrosome staining or electron microscopy is required. The midpiece is the metabolic powerhouse of the sperm. A helical sheath of mitochondria wraps tightly around the axoneme, the microtubule-based core of the flagellum, in this region, providing the adenosine triphosphate required for flagellar beating. The axoneme itself is a 9+2 arrangement of microtubule doublets, with inner and outer dynean arms that convert adenosine triphosphate into the sliding motion that drives flagellar bending. The principal piece and end piece extend the flagellum, tapering as they go. Here is the critical paternal contribution that is often underemphasized. The sperm contributes the centriole to the zygote. After fertilization, this paternally derived centriole duplicates and nucleates the first mitotic spindle of the embryo. Sperm with defective centrioles can fertilize oocytes normally, but the resulting zygote cannot organize a functional first mitotic spindle, leading to developmental arrest that may not be detected until the following day of culture. This is a mechanistic explanation for some cases of fertilization without subsequent embryo development that might otherwise appear mysterious. Having examined the oocyte and sperm as individual cells, let's follow what happens when they unite and consider the intracellular architecture of the early embryo that emerges. The moment of fertilization marks a profound transition. The oocyte's carefully organized cytoplasmic machinery now becomes the operational environment of the zygote, and then of each successive generation of blastomeres. Understanding what the early embryo inherits and what it must generate for itself is fundamental to interpreting embryo development and predicting competence. The zygote inherits its entire cytoplasmic machinery from the oocyte. Organelles, the cytoskeletal network, maternal messenger ribonucleic acids, proteins, and regulatory molecules. All of these are oocyte-derived. The paternal contribution at this stage is essentially limited to the genome, the centriole, and a handful of sperm-specific proteins that contribute to egg activation signaling. This inheritance has a critical implication. The quality of the oocyte's cytoplasm at the time of fertilization is the primary determinant of early embryo developmental capacity. The embryo does not start fresh, it starts with whatever the oocyte provided. Mitochondria deserve special attention here. Mitochondria do not replicate during early cleavage divisions. This is a deliberate feature of early embryo biology. The mitochondrial population is partitioned between blastomeres as they divide, rather than being newly synthesized. This means that the total adenosine triphosphate generating capacity is fixed at the time of fertilization, and it decreases per cell with each division as mitochondria are distributed among progressively more blastomeres. This creates a genuine energetic challenge for embryos with initially low or dysfunctional mitochondrial pools. The early cleavage divisions from zygote to morula are characterized by symmetric distribution of organelles and cytoskeletal components, a requirement for equal, synchronous cell division. Any asymmetry in organelle distribution or cytoskeletal organization tends to produce unequal blastomere sizes, which is an observable morphological abnormality that flags potential intracellular dysfunction. Compaction at the morula stage requires epithelial cadherin-mediated cell-to-cell adhesion, a molecule that is physically linked to the actin cytoskeleton through catenine proteins. Blastocele formation then requires active sodium-potassium adenosine triphosphatase activity in the trophectoderm plasma membrane to pump sodium ions into the intercellular space, drawing in water osmotically to form the fluid-filled cavity. Finally, lineage segregation into the inner cell mass and trophectoderm depends on differential gene expression driven by asymmetric intracellular signals. The embryo is not passively drifting toward implantation. It is actively constructing its own cellular hierarchy. With this mechanistic understanding in place, we can now make the critical clinical connection. How does intracellular organization predict developmental competence and in vitro fertilization outcomes? Here we arrive at the point where basic cell biology becomes directly actionable in the clinical laboratory. The question is this: what is the relationship between intracellular architecture and the outcomes we measure, fertilization rate, blastocyst formation, implantation, live birth? The answer is both direct and profound. Mitochondrial function is perhaps the most extensively studied intracellular predictor of assisted reproductive technology outcomes. Oocytes and embryos with higher mitochondrial membrane potential, a proxy for adenosine triphosphate production capacity, show significantly higher fertilization rates, better blastocyst formation rates, and improved implantation success. This makes biological sense. Every step of early development is energy-intensive, from spindle assembly to compaction to trophectoderm differentiation. Cells that cannot generate sufficient adenosine triphosphate will fail at one or more of these steps. Meiotic spindle integrity is directly coupled to chromosome segregation fidelity. The spindle apparatus must capture all 46 chromosomes at metaphase 2 and segregate exactly 23 to the first polar body and 23 to the mature oocyte. Any defect in spindle assembly, microtubule instability, aberrant motor protein function, centrosome dysfunction, increases the probability of chromosome missegregation and aneuploidy. And aneuploidy is the single largest contributor to implantation failure and early pregnancy loss after in vitro fertilization. Notice the relationship illustrated here between spindle integrity and aneuploidy risk. This is one of the most clinically important connections in all of reproductive biology. Cortical granule distribution and endoplasmic reticulum organization determine the efficiency and completeness of the block to polyspermy. In polyspermic oocytes, the cytoplasmic architecture has failed to implement the Zona reaction effectively, leading to triploid embryos, which are developmentally lethal in almost all cases. At the embryo level, symmetric cleavage and regular blastomere size are direct reflections of intact cytoskeletal function. Fragmentation reflects abnormal cytokinesis, typically attributable to cytoskeletal or cell cycle checkpoint dysfunction. Time-lapse morphokinetic parameters, the timing of first cleavage, the duration of cell cycle stages, the timing of blastulation, serve as indirect proxies for intracellular competence. And oocyte dysmorphisms visible at the time of intracytoplasmic sperm injection, cytoplasmic granularity, vacuoles, dark or uneven cytoplasm, are morphological markers that reflect underlying organelle dysfunction. They are diagnostic signals, not decorative details. Learning to read them accurately is one of the core practical competencies of clinical embryology. Understanding how intracellular architecture can be challenged helps us appreciate why optimizing every step of the assisted reproductive technology process matters so fundamentally. Let's turn to that now. Every single step of the in vitro fertilization process imposes measurable stress on the intracellular architecture we have been discussing. This is not a reason for alarm. It is a reason for rigorous, evidence-based protocol optimization. Let's work through the assisted reproductive technology workflow and identify specifically where each step challenges cell organization. Oocyte retrieval exposes cumulus oocyte complexes to rapid pressure changes during aspiration and to temperature fluctuations between the follicular environment and the incubator. The meiotic spindle is acutely temperature sensitive. Even brief cooling to room temperature can cause microtubule depolymerization, spindle disruption, and chromosome scatter. This is why heated stages are non-negotiable in the embryology laboratory, and why the time between aspiration and placement in the incubator should be minimized as a matter of routine practice. Denudation, the mechanical and enzymatic removal of cumulus cells prior to intracytoplasmic sperm injection, removes the oocyte from its extracellular support environment. Cumulus cells provide metabolites, growth factors, and gap junction-mediated signals that support oocyte metabolism. Their removal is necessary for intracytoplasmic sperm injection, but should be performed efficiently to minimize the oocyte's time without this support. Intracytoplasmic sperm injection itself introduces a fundamentally different mechanical interaction compared to natural fertilization. Direct injection of the sperm bypasses zona penetration and plasma membrane fusion, and the injection pipette physically penetrates the oocyte cortex. Done correctly, intracytoplasmic sperm injection is remarkably well tolerated. Done poorly, with excessive force, injection into the spindle, or failure to immobilize the sperm properly, it can introduce cortical damage, cytoskeletal disruption, and even mechanical trauma to the meiotic spindle. In vitro culture exposes embryos continuously to conditions that differ from the in vivo environment. Suboptimal oxygen tension generates reactive oxygen species that damage mitochondrial function and deoxyribonucleic acid. Inappropriate potential of hydrogen disrupts cytosolic enzyme activity. Osmolality shifts stress the plasma membrane and cytoskeletal network. Temperature fluctuations disturb microtubule dynamics. The development of sequential culture media, time-lapse incubators that minimize disturbances, and low-oxygen culture systems has been driven precisely by the understanding that embryos have specific intracellular sensitivities. Cryopreservation via vitrification prevents ice crystal formation. Ice crystals that would physically rupture membranes, cytoskeletal filaments, and organelle boundaries, but achieves this through rapid dehydration and exposure to concentrated cryoprotectants, which impose their own osmotic and chemical stress. The remarkable survival rates achieved with modern vitrification protocols reflect how much we have learned about these intracellular vulnerabilities and how precisely we can design protocols to protect against them. Knowing that intracellular architecture underpins oocyte and embryo quality, let's now consider how we assess that architecture in clinical practice through the lens of oocyte morphology. Clinical embryology is fundamentally an exercise in inference. We cannot directly visualize the intracellular architecture of a live oocyte, but we can read the morphological signals that the cell presents to us and interpret them through the lens of cell biology. This slide maps the major oocyte dysmorphisms to their likely intracellular origins. A healthy, mature metaphase II oocyte with intact intracellular organization presents a characteristic appearance, a clear, even cytoplasm without aggregates or vacuoles, a visible and intact zona pellucida, a first polar body present in the paravital space, and dimensions appropriate for the species. This appearance reflects normal mitochondrial distribution, intact endoplasmic reticulum network architecture, correctly positioned cortical granules, and a functional meiotic spindle arrested at metaphase II. Cytoplasmic granularity, a coarse, heterogeneous texture to the cytoplasm. is one of the most common oocyte dysmorphisms. Mechanistically, it most likely reflects abnormal mitochondrial clustering, rather than being distributed homogeneously throughout the cytoplasm. The mitochondria have aggregated into focal clusters, producing areas of increased optical density. It can also reflect abnormal endoplasmic reticulum aggregation. Either way, the granular appearance is a reliable indicator of reduced developmental potential. Oocytes with centrally concentrated granularity consistently show lower fertilization and blastocyst formation rates in clinical studies. Vacuoles are membrane-bound, fluid-filled inclusions in the cytoplasm. Their presence can indicate endoplasmic reticulum stress, a state where the endoplasmic reticulum is overwhelmed by misfolded proteins and responds by forming abnormal membranous structures, or dysregulated autophagy, where the normal process of organelle turnover has become pathologically active. Small numbers of small vacuoles may be inconsequential, but large or numerous vacuoles, particularly those that persist after fertilization, are associated with poor developmental outcomes. Smooth endoplasmic reticulum clusters are among the most clinically significant oocyte dysmorphisms recognized by the European Society of Human Reproduction and Embryology. These are large, disc-like aggregates of smooth endoplasmic reticulum visible as a refractal structure in the cytoplasm. They are specifically associated with calcium dysregulation. The endoplasmic reticulum architecture that supports normal calcium oscillations at fertilization has been disrupted, and they are flagged in European Society of Human Reproduction and Embryology guidelines as warranting careful consideration before transfer of resulting embryos. Refractal bodies are dense, granular inclusions that represent accumulations of oxidized lipid and protein. Their presence signals oxidative stress within the oocyte, a state of reactive oxygen species accumulation that damages organelle membranes and deoxyribonucleic acid. Each of these dysmorphisms tells a story about intracellular health. The same interpretive approach applies to embryo morphology. Let's see how the intracellular principles we've established translate to embryo quality assessment. Embryo morphology grading is the primary clinical tool we have for assessing developmental competence in the laboratory, and every morphological feature we assess has a specific intracellular mechanism behind it. Understanding those mechanisms allows you to move beyond memorizing grading criteria to genuinely understanding what the embryo is telling you. Fragmentation, the blebbing of enucleate cytoplasmic fragments during cleavage, reflects abnormal cytokinesis. The contractile ring, which is normally formed by actin and myosin at the cell equator and gradually constricts to separate daughter cells, has failed to operate correctly. Minor fragmentation below 10 to 15% is commonly observed, and its clinical significance is modest. Extensive fragmentation above 25 to 30% is associated with significantly reduced implantation rates, correlating with cumulative cytoskeletal dysfunction. Multinucleation, the presence of two or more nuclei within a single blastomere, results from a different failure mode. The nucleus has divided but the cytoplasm has not, or nuclear division has occurred asynchronously and produced extra nuclei. Either mechanism implicates the cytoskeletal and cell cycle checkpoint machinery, specifically the spindle assembly checkpoint that should prevent nuclear division from completing until all chromosomes are correctly attached to the spindle. Multinucleated blastomeres at day 2 or day 3 are a reliable marker of reduced developmental potential and increased aneuploidy risk. Unequal blastomere size at the 2-cell or 4-cell stage reflects asymmetric cytokinesis. The cleavage plane was not correctly positioned at the cell equator, resulting in blastomeres of different volumes. This can be caused by aberrant spindle positioning, defective motor protein function, or compromised cytoskeletal mechanics. Developmental arrest, the failure to progress beyond a specific developmental stage, often reflects cumulative intracellular failure. Mitochondrial insufficiency means insufficient adenosine triphosphate for energy-intensive transitions between stages. Proteasomal dysfunction prevents normal cell cycle progression by allowing accumulation of inhibitory proteins. And maternal messenger ribonucleic acid depletion removes the instructions needed to synthesize critical proteins. Arrest at the 2 to 4 cell stage in particular often implicates a failure at the maternal to embryonic transition. At the blastocyst stage, inner cell mass and trophectoderm grades reflect successful lineage segregation, tight junction formation, and active fluid transport, all dependent on cytoskeletal integrity, cell-to-cell adhesion molecules, and plasma membrane ion transport function. The European Society of Human Reproduction and Embryology and the alpha scientists in reproductive medicine grading systems translate all of these intracellular events into standardized morphological scores that allow communication and comparison across laboratories. We have now covered all the content of this session. Let's bring everything together with a comprehensive summary of the key messages. We have covered substantial ground in this session, and before we close, let's work through a structured recapitulation of the essential messages. These are the conceptual pillars you should carry forward into every subsequent topic in this series. First, eukaryotic cells are compartmentalized systems. Each organelle maintains a specialized microenvironment that enables biochemical processes that could not occur in an unpartitioned space. Compartmentalization is not a structural luxury. It is the fundamental organizing principle that makes complex cellular life possible. Second, cytoplasm, cytosol, and organelles are distinct terms with distinct meanings. Cytoplasm encompasses the entire intracellular space, excluding the nucleus. Cytosol is the aqueous fluid phase. Organelles are the specialized structures, membrane-bound and non-membrane-bound, within the cytoplasm. These distinctions matter for understanding where cellular processes occur and why local disruptions have selective effects. Third, membrane-bound and non-membrane-bound organelles each make essential contributions to gamete and embryo function. The nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and endosomes collectively handle energy production, protein synthesis, calcium homeostasis, waste management, and vesicular trafficking. Ribosomes, centrosomes, proteasomes, and processing bodies contribute equally to protein synthesis, cell division, protein degradation, and messenger ribonucleic acid regulation. Fourth, the cytoskeleton is the structural and transport backbone of the cell. Its 3 filament systems, actin, microtubules, and intermediate filaments, together position organelles, transmit forces, drive cell division, and enable directed intracellular transport through motor proteins. Cytoskeletal integrity is required for spindle assembly, cytokinesis, cortical granule positioning, and compaction, every one of which is clinically observable. Fifth, the oocyte provides essentially all cytoplasmic machinery for the early embryo. Mitochondria, maternal messenger ribonucleic acids, ribosomes, cytoskeletal elements, cortical granules, and the endoplasmic reticulum calcium store are all oocyte-derived. The paternal contribution at this stage is genomic, centriolar, and activating. The intracellular quality of the oocyte at fertilization is therefore the primary determinant of early developmental potential. And sixth, assisted reproductive technology procedures impose measurable stress on intracellular structures at every step. Temperature changes disrupt spindle integrity. Osmotic stress challenges membrane stability. Reactive oxygen species accumulation impairs mitochondrial function. Optimized protocols are designed specifically to protect the intracellular architecture upon which competence depends. This understanding should inform every protocol decision you make in the laboratory. With this comprehensive foundation in cell organization firmly established, we are ready to move outward from the cell's interior to examine its boundary layer. Join us for Part 2, the plasma membrane, structure, and lipids. Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.